So hello, everyone. It is my great pleasure to introduce the speakers of today's webinar, my very good friend and colleague, Dr. Eric Vivier. So Eric graduated from the famous veterinary schools in Maison Alfort, which is close to Paris, and before doing a PhD in immunology in Paris 11. He then left France to do a postdoctoral fellowship as a Fogarty International Research Fellow at Harvard Medical School with Paul Anderson and Stuart Schlossman at the Dana-Farber. And in 1993, he was recruited back in France at the CIML, which is Center of Immunology at Marseille-Lumini, as an assistant professor, where he quickly rose to the rank to become the director of the center from 2008 until 2017. He is currently a professor of immunology at Aix-Marseille University and the Marseille Public Hospital. He's a member of the French National Academy of Medicine and of the famous Institut Universitaire de France. Eric made major contribution to our understanding of the development and function of natural killer cells and innate lymphocytes, both in mice and human. He identified key inhibitory receptors that control the function of innate lymphocytes and developed therapeutic strategy to harness their immune effector function against cancer. He's the author of more than 300 publications in immunology and ranks among the world's most cited researchers. He has been awarded several prizes and honors, including the European Federation of Immunological Society Award and the Grand Prix Charles Oberling in oncology. He's also a Chevalier de la Légion d'honneur. Eric is also one of the founders of Marseille Immunopole, which is an immunology initiative that integrates both basic science to further therapeutic innovation, but also industrial partnership to enhance immunotherapy strategy in the clinic. In 2018, while being one of the finalists to lead the Pasteur Institute in Paris, Eric declined the offer and instead accepted to become the chief scientific officer of Innate Pharma, a biotech that he founded a decade earlier. To really harness the potential therapeutic function of innate cells that he was working on, and now has become really a global leader in cancer immunotherapy. But what I find quite unique about Eric is, like, despite his many accomplishments, and despite all these titles, he's one of the most generous and passionate immunologists I know. He cares deeply about education, and this is why he's here today, really to share his journey path with us. And he's really passionate also about bringing immunology to the clinic, as you will hear today. So it's really an honor to introduce you today, Eric, and I'm very much looking forward to your talk. Thank you.
Well, thank you very much, Miriam. It's really my honor to be with you today. And I hope that your kind introduction will be, that I won't be a disappointment for everybody on the webinar. Okay, so thanks again for this nice introduction. So what I'll try to do today is indeed to review some stories that we had recently regarding the role of innate immunity, and in particular trying to focus our discoveries into new treatments for patients. And as you will see, we have been able also to propose a repurposing of some molecules that we initiated in cancer in the context of COVID-19. And at the very end, I have a surprise for you. Okay, so I will... let me do this now. Okay. So this is my disclosure slide. So as Miriam told you, actually 21 years ago, I had the privilege to co-found a company called Innate Pharma, which is both in France and in the US. And the part of the work that I will be presenting is actually made at Innate Pharma. So I will start by this very obvious slide that everybody has shown, some version of this slide at least, to realize that it's really a good time for an immunologist. As we know that one can translate the findings of basic immunology into innovative treatments for patients with different kinds of cancers. And this is particularly true for immune checkpoint inhibitors. But as you see here with this green line and this question mark, we are not yet there. Unfortunately, in many cancer types, despite the success of immunotherapy, there are resistance to this treatment. And one has to understand this resistance to immune checkpoint inhibitor or other immuno-oncology treatments in order to increase the fraction of patients sensitive to these treatments. And at the same time, we have to be extremely cautious about the toxicity which is linked to this treatment. So this is basically our mission. This is a mission that we gave to ourselves, and with a special angle, which is the one of innate immunity. Why? Well, for obvious reasons. It's obvious that, in particular, in the context of immuno-oncology, T cells are central to the control of cancer. But we know from decades of immunology that T cells are not autonomous in what they do. They need help from other cells, namely the cells of the innate immune system, to be effector cells and to be able to control cancer. So this is why we have actually refined or redesigned, in a way, the famous cancer immunity cycle pioneered by Dan Chen and Ira Mellman to introduce the key steps at which the innate immunity is so important in actually mounting an adaptive immune response. And this is what we are trying to do. And with a particular focus on this kind of lymphocytes, which are called innate lymphoid cells. So as you probably know, lymphocytes are not only T cells and B cells, but there are also some innate lymphocytes, innate lymphoid cells. The prototypical example being natural killer cells, NK cells, here represented on the right-hand side in an interaction with a tumor cell. So as you probably know, these innate lymphoid cells are coming in 5 different flavors, but for the sake of time for today, I will just focus on natural killer cells and at the end, ILC1s. So why NK cells? So this is another version of this innate immunity cycle in which we highlighted the role of NK cells. We know that NK cells have the propensity to recognize a vast variety of tumor type. And this is true, obviously, in humans, but this is also true in preclinical models, in particular in the mouse. And this is extremely useful for the dissection of the biological function of NK cells. But as you know, beyond this capacity to kill tumor cells, NK cells can also be some kind of a spark at the tumor bed to initiate a multicellular immune response that will finally end or culminate into a protective T cell immunity against cancer. And this is highlighted here when you see that NK cells can actually produce an array of cytokines such as XCL1, CCL5, or class 3 ligands that really help dendritic cells to do what they do, meaning to promote a T cell immunity. At the same time, NK cells can also produce gamma interferon, which obviously help in the shaping of the adaptive immune response. So you see that here, by trying to target or to harness NK cells, we are not only hoping to double the tumor, which will probably not happen, but we are trying to initiate a multicellular immune response. So that said, we are concentrating our approaches in innovative anti-cancer treatments using antibodies. And so you know that there are 2 kinds of antibodies, broadly speaking. On the left-hand side, you have these blocking antibodies, the prototypical example being anti-PD-1, anti-PD-L1, that block inhibitory interaction between a T cell and a tumor cell, unleashing the immune effector cell, in particular T cells, and enable them to kill tumor cells. But on the other side, on the right-hand side of this slide, you see also that there are these cytotoxic antibodies, which in the context of cancer can target tumor antigen. And we know that these cytotoxic antibodies need cells expressing receptor for the Fc fragment of the antibodies in order to being able to enable ADCC, antibody-dependent cell cytotoxicity, or ADCP for anti-dependent cell phagocytosis. So the first kind of set of data that I would like to present to you today after this introduction is really about the cytotoxic antibody technology. How can we actually boost these cytotoxic antibodies, which are, as represented here, are recognizing a tumor antigen? So one needs an FcR positive effector cell, as I said, but can we try to manipulate this immune interaction so that these cytotoxic antibodies will be very effective? So one way of doing this is actually to combine the two kinds of antibodies I just told you about. So in green, you have the cytotoxic antibody that recognizes the tumor antigen, but in blue, you have a blocking antibody. And here, I took the example of Monalizumab, which is an antibody that has been developed by Innate Pharma, is not developed by AstraZeneca, which actually blocks the interaction between an inhibitory molecule expressed on NK cells and T cells, which is called NKG2A, and its partners is cognate ligand, which is a non-classical MHC class 1 molecule called HLA-E in humans and QA1B in the mouse. So it's really a nineteen-bearing molecule, this NKG2A receptor, meaning that it can recruit through intracytoplasmic tyrosine-based inhibitory motif, ITIMs, the protein tyrosine phosphatases SHP1 and SHP2, and hence block the activation of the effector cells. So what we were thinking is actually try to boost the activity of cytotoxic antibodies by combining the treatment with Monalizumab. And this is what we have been doing in an array of preclinical data, but I would just like to focus on a clinical trial, a phase 2b clinical trial that we have performed in patients with head and neck cancer. So what you can see here is a waterfall plot in which I remind you each histogram represents the best increase, unfortunately, or the best decrease of a tumor lesion. And here, this is a cohort of patients who received a combination of cetuximab, which is an antibody that recognizes EGFR, which, as you know, can be overexpressed on tumor cells, and Monalizumab. So it's really the treatment which is represented on the left-hand side of the slide with a green antibody, which will be cetuximab, and the blue antibody, Monalizumab. And as you can see from the waterfall plot on the right-hand side, there really is an advantage of treating these patients with a combination of Monalizumab and cetuximab. And this is why now Monalizumab has been developed by AstraZeneca to enter a phase 3 clinical trial in this indication. So this is a cartoon that tells you more about the use of Monalizumab, because for the time being, I just told you about the left-hand side of this slide, when you see again the combination of cetuximab and Monalizumab. But if you pay attention, on the right-hand side, there are not only NK cells, but also T cells, because very importantly, a large fraction of PD-1 positive T cells at the tumor bed co-express NKG2A. So hence the idea actually of combining the blockade of PD-1, of the PD-1 PDL1 axis, I should say, with the blockade of the NKG2A HLA-E axis. And this is what is being currently tested in clinical trials, the combination of anti-PDL1 or anti-PD-1 plus Monalizumab in some cancer indications. And here you have quoted the paper in which most of the preclinical data have been reported. Now I would like to switch gear to another attempt to boost cytotoxic antibodies, because one can try to harness the effector function of immune cells by blocking inhibitory molecules, as I've been telling you for Monalizumab. But one can try also to boost the stimulating molecule, the activating receptor expressed here on NK cells. So this is what we have been doing by generating what we call the NKp46 NK cell engagers, which are basically totally synthetic molecules built on the beauty of the antibody fragment Lego, in which we can reconstitute here an antibody with actually trifunctional capacities. And in the movie that you will see, and I will ask Anna to actually run the movie, you will see what we have been doing. starting with an anti-NKP46 that recognize the tumor antigen. And hence, you can understand here the trifunctional capacity of these NK cell engagers. Can I go back to the slides? Here we go. Okay, so this is another version of what we are attempting to do. Here again, you see the NK cell engager bridging the tumor cell and the NK cell. And one of the key features of this NK cell engager through the particular geometry of this molecule is to co-engage simultaneously CD16 and NKP46. And as you will see, this is extremely important for the activity of this molecule. And what we are trying to do is to kill the tumor cell, obviously, but not only that, what we are also trying to do is to induce the secretion of cytokines and chemokines by NK cells, such as XCL1, CCL5, and STRAT3 ligand. And here, the idea is actually to boost dendritic cells for their activity to actually trigger a T cell activity against the tumor cell. So some raw data now. What you can see here is a comparison of 3 flavors of this NKP46 NK cell engager. There's a color code in purple on the left-hand side. You have the Fc silent portion of this NK cell engager. So it's really a bispecific antibody targeting NKP46 on one side and the tumor antigen on the other side. In the middle, in red, you have the wild-type version of this NK cell engagers with the Fc wild-type portion of this human IgG1. And in blue, on the right, you have actually an enhanced version of the Fc portion of human IgG1 so that it binds CD16 with higher affinity. And what you can see is that when you increase the affinity of this NK cell engager for CD16, you increase the efficacy of these NK cell engagers. And this is here testing primary human NK cells with 3 different targets, with 3 different tumor antigens, CD20, CD19, or EGFR. What is also very important, I think, and this is on the very right-hand side of this slide, is that when you compare within the limit of this preclinical setting, the activity of the NKP46 NK cell engager with antibodies that have been approved in the clinics, such as obinutuzumab or rituximab for CD20 or cetuximab for EGFR, you can appreciate that actually the NK cell engagers are much more effective than this clinically approved antibody. So this is extremely important for the development or for the further development of this molecule into the clinic. So as I said, the geometry of the molecule is extremely important, and this is represented here on this data slide. When you see 2 different settings, on the left-hand side, the upper graph, you see the NK cell engager that engages simultaneously CD16 and NKP46 with the red head for CD16 and the green head for NKP46. And this corresponds to the red curve on the right-hand side. So very effective molecule in terms of inducing the lysis of the tumor cell. Now, when you compare this to another setting in which we have a bispecific antibody, green and orange, that targets NKP46 and CD20, added to an antibody against CD20, which would be the red and orange, you can see that on the right-hand side that we are actually losing 2 logs of efficacy. So really the fact that we can co-aggregate CD16 and NKP46 on a single NK cell is key to the activity of this cell, and this paper has been published as quoted here. So basically what I've been telling you, and this is a graph in which we've been trying to review what has been done to boost the activity of anti-tumor antibodies. One can combine these cytotoxic antibodies to immune checkpoint inhibitors with the example of Monalizumab, this is panel C, or one can create a new agonistic antibody by using the Lego of the antibody bioengineering technology, and this is the NKp46 NK cell engagers, and others have also tried to generate NK cell engagers with different moieties. For example, introducing IL-15 in this strike molecule pioneered by Jeff Miller in Minnesota, or others have been also developing bispecific antibodies engaging CD16 and the tumor antigen. I should say that we choose NKp46 to be associated to CD16 for a peculiar reason, is that NKp46 is really well expressed on NK cells at the tumor bed, which is different as compared to NKG2D, for example, which can be readily downregulated at the tumor bed, and other activating NK cell receptors as well. So now we'd like to switch gears and tell you what we've been trying to do in the context of COVID-19. And the idea was actually to try to take advantage of the knowledge that we could accumulate over the years in the manipulation of the innate immune system in the context of patients presenting severe COVID-19 pneumonia. So as we know, there are unfortunately only a few percentages of SARS-CoV-2 infected individuals that can develop acute respiratory distress syndrome, so ARDS, but we have to treat these patients, and this is urgently required. We know that the immune system is playing a dual role in COVID-19. The immune system can obviously contribute to virus elimination, but it can also contribute to ARDS development, which can be seen as some kind of overt inflammation. So in Explore COVID-19 program, we set up a very large translational study to try to characterize the immune response occurring in patients having been infected with SARS-CoV-2, and as you will see, in 4 groups of people from ARDS to polysymptomatic patients. And the idea behind this was to try to repurpose approved immunomodulatory molecule or candidate drugs, which are been already tested in clinical trials for the benefit of COVID-19 patients. So this is a very busy slide, but basically what you have to remember is on the left-hand side, where you see the different groups of patients from who we can get samples, blood samples or bronchoalveolar lavage fluid, and I'll come back to that. So we've been harvesting samples from healthy controls in black, and the color code will be obviously maintained in the data slides. We had also access to patients with polysymptomatic COVID-19, patients with severe pneumonia, but not in ARDS, and these patients will be in green. And finally, a group of patients with acute respiratory distress syndrome that necessitated ICU. So as many groups over the world, we have been able to document the lymphopenia, which is associated to the severity of the disease. And this lymphopenia is not only a T cell or B cell lymphopenia, but it is also an NK cell lymphopenia, as you can see here. So we've been obviously looking at markers of inflammation, such as CRP, but also at the so-called cytokine storm, focusing here on IL-6, CCL4, CCL2, and CXCL9. And that's true that in the majority of patients with ARDS, we have an increase in circulating IL-6, CCL4, CCL2, CXCL9. But you can also appreciate that there is a substantial fraction of patients with no detectable increase in these cytokines. And in the interest of time, I just cut a lot of slides, but what we've been doing is just, is not only to take one blood samples, but it was a longitudinal study across 3 different time points. And when I tell you that there are some patients with no detectable increase in IL-6, although they were in ARDS due to SARS-CoV-2 infection, it's not only true at the time of inclusion in the cohort, but it's also true 4 days and 10 days later. So there is really a fraction of patients with no detectable cytokine storm, although they are in ARDS. So we've been focusing on the complement. And why is really obvious, because again, as at Innate Pharma, we are developing molecules targeting the innate immune system against cancer. We had access to some of these molecules, in particular antibodies that can interact with the complement pathway. So it was obvious to us to look at this activation of the complement in the idea of repurposing eventually some of these molecules. And so, as you know, the complement cascade can be activating using 3 different pathways: the classical pathway, the lectin pathway, and the alternative pathway. But I would like you to pay attention to one of the molecules of the complement cascade, which is called C5a, because C5a is really an outlier in this complement cascade for one particular reason. All the molecules of the complement cascade participate to the immune function of the complement. And I remind you that the complement is actually very important to cope against a certain kind of microbial infection, in particular bacterial infection. So we need the formation of the membrane attack complex, the MAC, which is the aggregation of C5b to C9. But again, there is an outlier, which is C5a, because C5b does not participate to the formation of the MAC. It's just a byproduct, so to speak, of the cleavage of C5 into C5b, which again participate to the MAC formation, and C5a itself. So C5a has been identified initially as an anaphylatoxin, such as C3a, but C3a is really upstream of the complement cascade, and C5a is downstream of the complement cascade. And this C5a has been shown in many different conditions to be a pro-inflammatory molecule, in particular in the context of viral infection, and in particular in the context of viral-induced pneumonia. As you can see on this slide, in preclinical models, in both mouse and non-human primates, using H1N1, H5N9, N1, or H7N9, at each time one can see there is a drastic inflammation in the lung, which really mimics what can be seen in COVID-19 ARDS. And in these particular studies, the blockade of C5a could actually inhibit the inflammation in the airways, inflammation which is due to the infiltration of the lung parenchyma by myeloid cells, in particular neutrophils and macrophages. And you can really see that on the right-hand side, when you see in this non-human primate model an infection with H7N9, and you see in the control column a staining for macrophages or neutrophils in panel G and I, and you see some brown colors that really illustrate the infiltration of this lung parenchyma by macrophages or neutrophils. Now, when you treat the non-human primates with an anti-C5a antibody, you can actually block entirely the infiltration, and you can actually block the inflammation of the lung as well. So it was really tempting to us to look at C5a in severe COVID-19 patients, and as you can see here, sure enough, there is a very obvious increase in circulating C5a, which is totally commensurate to the severity of the disease. So it looks like CRP, for example. And you see that this level of C5a is actually maintained both in severe pneumonia in green and in ARDS patients in red. And now you look at the right-hand side of this slide, and at not only at the time of inclusion in the cohort, but also at 4 days and 10 days, you really can see that there is a sustained high level of C5a in the serum of these patients. So we looked at other markers of the complement pathway activation. In particular, we looked at deposition of C5b9. And we looked precisely at the vascular compartment, because more we know about COVID-19, inflammation, and severe pneumonia, more we know about the fact that this severe COVID-19 is actually a vascular problem. There is an inflammation of the endothelium. There is also thrombi almost everywhere. And this is an important point for treating these patients. So here, as you can see, there is a beautiful, quote-unquote, beautiful lesion of obliterating endarteritis when you actually don't see the lumen of the arteries anymore, but because it's totally clogged with cells, and not only cells, but extracellular protein. And what you can see on the right-hand side is that there is a deposition of the C5b9, so the aggregation of C5b, C6, C7, C8, and C9, which is occurring there, telling us, witnessing the fact that there is a local activation of the complement pathway. So how does C5a work? Well, C5a works through two kinds of receptors, 7 transmembrane PRAS receptors, C5AR1 and C5AR2. We know not much about C5AR2, I should say, the biology of which remains kind of blurred. All we know is that C5AR2 might rather be coupled to beta-arrestin, so rather an inhibitory molecule in a nutshell. Now, C5AR1, we know quite a lot about C5AR1, and C5AR1 is really an activating receptor, which is also a marker of the myeloid cell compartment. It's expressed on neutrophils, macrophages, monocytes, dendritic cells. So in the interest of time, I just cut here some important results. So as I told you, C5AR1 is really a marker of myeloid cells in normal conditions, but it's also very highly expressed during COVID-19 on these myeloid cells in the blood, so remains high. So if there is C5a, there is the possibility of an effect of C5a because the receptors for C5a are still there. And we could show by taking PBMCs from COVID-19 patients and activating them with LPS or R848, which is probably a better inducer here because R848 is activating TLR7, TLR8, such as RNA viruses. But so when we add C5a to this culture, we can increase the production of inflammatory cytokines such as IL-6, TNF, and CCL2. So, but as we've been looking at severe COVID-19 pneumonia, it was very important for us to look at the lung, so we could have access to bronchoalveolar lavage fluid. And sure enough, you can see that also in the BALF, bronchoalveolar lavage fluid of these severe COVID-19 patients, you see, and here's just patients with ARDS, acute respiratory distress syndrome. You can see a very high level of C5a, very high level of IL-8 as well, and CXCL9 and CCL2. And you can notice there is not much of IL-6 in this bronchoalveolar lavage fluid. Now, on the right-hand side, what you see that neutrophils and monocytes express C5AR1, as I told you. So we've been further documenting what was happening in the lung by looking at immunofluorescence and the infiltration of the lung using some kind of myeloid cell marker. So obviously C5AR1, but also CD163 and CD68. And you can see that there is indeed in COVID-19 a toxic lung specimen, an infiltration with this myeloid cell that express C5AR1. So we could also analyze single-cell RNA-seq data from lung cells, from healthy controls on the left-hand side of the UMAP plot, or ARDS COVID-19 patients. So you can see that we can actually annotate the different cluster of cells in what we call subset A or subset B. So basically, those are subset of myeloid cells. I don't want to go into the detail, but just specifically, we looked at, again, the genes encoding for these inflammatory cytokines, and sure enough, in the subset B, you have an increase in the transcripts encoding for this inflammatory cytokine. So there is a major change in the myeloid compartment infiltrating severe COVID-19 patients' lung. So now that we know that there is C5a in the serum, there is C5a in the lung, there is also an infiltration of cells expressing C5AR1, and these cells can produce cytokines. It was obvious to see whether we can try to help these patients by blocking the pathway. So we had access to an antibody, which is called Avdoralimab, which is a human Fc-silent anti-C5AR1. And here's just one example of the activity of C5AR1. So here we are looking at the activation of neutrophils upon C5a treatment, and the marker that we have been using is just the induction of CD11b. So what you can see on the left-hand side, the panel A in black, open histogram is actually the induction of CD11b as compared to the gray histogram. What you see in purple is actually the total blockade of this induction when we treat the neutrophils, which are treated themselves by C5a using Avdoralimab. And on the right-hand side, you have a dose response of C5a using megadose of C5a. You can block entirely the induction of CD11b on fresh human neutrophils using Avdoralimab. So Avdoralimab is really a good reagent that can block the C5a, C5AR1 pathway. So here what we've been doing is, I think, very important, and it's probably what we can add in the description, the dissection of the pathology induced by SARS-CoV-2. Because our model is that the infiltration of myeloid cells within the lung is actually key to the inflammation occurring in these patients. And here what you see is the chemotactic activity of C5a. So if there is C5a in the lung, and I've been showing you that, then it makes a lot of sense that C5a could actually attract myeloid cells from the blood to the inflamed lung. And we've been mimicking this chemo attraction using Transwell assay. So here we are taking freshly isolated human neutrophils, and in a Transwell assay, you can see that C5a is really inducing a very nice migration. This is totally blocked by Avdoralimab. So we wanted to challenge this model in vivo using a model of acute lung injury. And we've been using a human C5AR1 knock-in mouse so that we can use the Avdoralimab antibody that blocks the human C5AR1 and not the mouse C5AR1. So these mice are very nice because there is a total and faithful replacement of the mouse endogenous C5AR1 by the human C5AR1. So when we instill C5a in these mice, there is a very nice induction of CD45 plus cells in the lung, and these cells are actually of the myeloid lineage, both neutrophils and monocytes/macrophages. There is also destruction of the lung parenchyma as seen by the release of albumin. So we've been obviously treating the mice using Avdoralimab, and as you can see here, one can totally block the inflammation induced in the lung by treatment with this blocking antibody. So basically, what we are proposing is that there is two different kind of disease upon SARS-CoV-2 infection. There's a non-severe COVID-19 in which the immune system is actually coping with this infection. We know that this is in part type 1 interferon dependent, so the immune system is happy Joe and is successful in controlling the virus infection. You don't have much of the activation of the complement. This is the left-hand side of this slide. This is an epithelial disease. As you know that the receptor for SARS-CoV-2 is ACE2, in particular expressed on type 1 on pneumocytes. Problem is then where there is a defect in this immune response, there is an hyperactivation of the complement. And this hyperactivation of the complement is leading actually to an endothelial inflammation. And because you know that I have no time to go into the detail, but what is important about the complement cascade, it's really as a crossroad between cytokine secretion, induction of cytokine secretion, but also induction of endothelial inflammation and prothrombotic state. So this is what we have been publishing very recently, and so unfortunately you will never see this cover of Nature because they took the paper, but they didn't take the cover. We are not complaining, by the way, and so but the idea here was to try to illustrate that avdoralimab in blue, like a rain, can actually extinguish the fire induced by SARS-CoV-2 in the lung of the patients. So these results were actually very consistent with very recent results published on the bioRxiv platform, where other colleagues, Ma et al., have reported that there is an increase in complement activation during severe SARS-CoV-2 infections. It's really distinctive, a distinctive feature, because these colleagues could actually compare with patients in ICU for non-COVID acute respiratory failure, and in particular induced by flu, and they didn't see as much of complement activation as one can see in the context of SARS-CoV-2 infection, and they could see markers of endothelial injury, such as angiopoietin-2, as well as hypercoagulability. So we do think that the activation of the complement is very important, and that there are means, at least through avdoralimab, to actually treat these patients. So this is where the Explore COVID-19 translational study became a clinical trial. And we call the clinical trial FORCE for COVID-19 elimination. And basically, this is the design of the clinical trial, which has been completed. So we are waiting for the analysis of this double-blind randomized phase 2 clinical trial, which has included 208 patients in 3 different arms, as you can see, from severe COVID-19 pneumonia, which did not need mechanical ventilation, up to ARDS patients, and we are obviously looking forward to the analysis of this clinical trial. So I'm not going over this conclusion because I've been telling you already, just in a nutshell, we do think that C5a is not only a marker of the inflammation occurring during COVID-19, but it's possibly a very important actor of the inflammation, in particular because of this activity of attracting this chemoattractant activity, allowing the recruitment of myeloid cells to the lung. So now I would like, if I have some time, to finish up with some results that are in press but not published, coming back to the roots, in a way, to the innate lymphoid cells, and highlighting the difference between NK cells and ILC1s. Because basically, NK cells and ILC1s can really look the same. They look alike. They are driven by Tibet. They can produce gamma interferon. They share many similar surface markers, but they are different. And I would like to highlight the developmental difference between NK cells and ILC1, in particular in the liver. So let's start by this slide, just to tell you that there is, in fact, some kind of blurriness about the definition of NK versus ILC1s, in particular in humans, and in different locations. Because I remind you that ILCs are mainly tissue-resident cells, in contrast to NK cells. But there is one location and one species in which it's absolutely clear it's the liver. The mouse liver actually contains basically half and half NK cells and ILC1. And one can discriminate NK cells from ILC1s using a very useful set of markers, which are beta-1 integrin, CD49A and CD49B. So in a nutshell, NK cells and ILC1 will be CD3 negative, lineage negative, I should say, but NK1.1 plus, NKp46 plus, both of them. But then NK cells would be CD49B plus, CD49A negative. And in contrast, ILC1 would be CD49A plus, CD49B negative. There are also some other markers that can differentiate NK cells from conventional NK cells from ILC1s. And you can see that in the B panel, in which in red, you see the markers which are expressed by ILC1s. So CD200R1 or TRAIL or CD69 are expressed by ILC1 and not by NK cells. As I told you, both ILC1 and conventional NK cells express the transcription factor TBET, but a major contrast, and this will be important in the end, conventional NK cells do express AOMS or AOMS, transcription factor, in contrast to ILC1s. There are also some difference in terms of effector function because NK cells are better interferon gamma producers as compared to ILC1s. And in contrast, ILC1s produce more TNF-alpha as compared to conventional NK cells. So with that in mind, we were interested to actually try to dissect this phenomenon, which is illustrated here. We were interested in collaboration with our colleagues and friends in Hefei, China. This is a group of Xingang Tian with Wei Peng. So we were interested, as I said, to try to understand what was going on in terms of ILC1 versus NK cell reconstitution in irradiated mice. Because in contrast to NK cells, which are not represented here, ILC1s are not very well at all reconstituted by bone marrow cells. And this is BMT that you can see here. You can see in percentages, you can see in number of cells. Really, the reconstitution is not good. But when you take fetal liver, it works quite well. So the idea was to try to challenge this a bit and maybe go with the adult liver. And to make a long story short, when I say we, is our colleagues in China and ourselves, could actually identify a population of what we call LSM cells, lean negative SCA1 plus MAC1 plus, which is present not only in the fetal liver, but also in the adult liver. And one of the features of this population of cells is that they can readily reconstitute ILC1 specifically in the liver. I see that I'm running out of time, so I will go very fast. But this is the point. So LSM cells can reconstitute preferentially ILC1 in the liver and not NK cells. So we could actually perform single-cell RNA-seq to understand the heterogeneity of this population of cells. I have no time to go into the details, but basically we could define another step in the differentiation from LSM cells to ILC1 with an intermediate cells, lineage negative, CD122 plus, CD49A positive. Again, I will be happy to take questions. I will run fast now. What was very important is that on the other side, once we have shown that ILC1s in the mouse liver were reconstituted by adult progenitors from the liver and not from bone marrow progenitors, we could show that this process was entirely interferon gamma dependent. Because using interferon gamma knockout mice or interferon gamma receptor knockout mice, one can actually very much attenuate the number or lower the number of ILC1 specifically in the liver of the mice. Now, when you inject interferon gamma in the mice, one can really increase ILC1 numbers. So we looked at what kind of cells could produce interferon gamma. And it's really not T cells. It's not B cells either. I mean, B cells do not produce much gamma anyway. But you see that it's not CD4, it's not CD8. So what kind of cells could actually produce gamma and being able to amplify the production of ILC1? So we took advantage of a mouse that we generated years ago, which is an NKP46 screen mouse that actually can genetically tag all NKP46 positive cells. So in short, ILC1s and NK cells. And to make here a long story short, we could show that when we get rid of gamma interferon using these CRE-mice. So we ablate the capacity of NK cells on ILC1 to make interferon gamma, as you can see here. There is actually a total phenocopy of what we see in the complete gamma knockout. So namely, the NKp46 ILCs themselves are producing the gamma, which is important for the differentiation and the development of liver ILC1. And previously, in collaboration with Steve Reiner, we could show that when we ablate EOMs using the same NCR1 CRE mice, we could actually really deplete NK cells, but we are not touching ILC1s at all. So we know that it's not NK cells, it's not the gamma interferon produced by NK cells, which is so important for the production of liver ILC1. So basically what we are proposing, and sorry I was going very fast here, is actually something which is interesting, is the fact that very much like the myeloid cells, which are derived for part of them from progenitors coming from the embryonic life, for instance, particularly the work of Myriam and in collaboration with Florent Ginoux. So similarly to that, we have a wave of ILC1 progenitors which come from the fetal liver, which seed the liver, and these progenitors will be still there during adulthood. And there is here some kind of a loop which is interferon gamma dependent, which is fed by ILC1s themselves. So the LSM cells are giving rise to these intermediate cells, which give rise to ILC1s, and ILC1s produce the gamma selectively in the liver. And the receptors for gamma are only on LSM cells in situ. So basically, there is a local development of liver ILC1s due to an interferon gamma dependent loop. So I'm going to stop here, and I'm going to thank first all the patients and the families, patients who have participated to the clinical trials that we are running or that we have been running. I would like also, obviously, to thank people of the lab and the funders, in particular, the European Research Council. Thank you very much for your attention.
Perfect. Thank you very much, Eric. I'm glad that you cut some slides because indeed you covered a fantastic area of immunology. Thank you very much for this talk. You had many, many questions, so I'm going to try to go through them, and there are questions about all the three different parts of your talk. I'm going to start with the first part, which is the anti-tumor immunotherapy, and there is this question from Pini Zuckerman asking whether can you attribute the effect of Monalizumab to T cells or NK cells?
Yeah, that's a very good point, and thanks for the question. It's impossible. It's impossible to attribute the effect of Monalizumab to either NK cells and CD8 T cells in patients. Obviously, in the mouse, it's easy, and we've done that. But in patients, it's not doable. And I'm not sure it will be, unless we will be treating patients with some kind of specific immune deficiency, for example, a T cell deficiency, which then obviously would be, if there is an effect, it would be due to NK cells. But otherwise, it's impossible because Monalizumab, and thanks for the questions, by the way, Monalizumab is, I think, the prototypical example of a new category of immune checkpoint inhibitor that we can call the broad spectrum immune checkpoint inhibitors, which are not only unleashing T cells, but unleashing many, many cells beyond T cells. And Monalizumab, again, will be unleashing both NK and CD8 T cells.
And this is why maybe it's a very important molecule in the clinic. I think the ability to target more than one, right?
Absolutely, absolutely.
Probably very beneficial to patients.
Absolutely.
Another question from Divan Sarhan is how does the trifunctional the trikes affecting NK cells in long-term culture? Will NK cells become exhausted?
Okay, we didn't see that. We've been, no, it's a very good point. I should say that for the experiment that we've been running so far, we didn't see any death or exhaustion of NK cells upon NK cell engager treatment. But that's maybe we didn't run the assay long enough. I don't know, but we didn't see such a thing like an exhaustion of NK cells induced by NKCE so far. What we see, though, and that I didn't mention, is an infiltration of NK cells in vivo as a tumor bed induced by NKCEs.
That's interesting. There is a question from Shubhada asking, does Fc gamma receptor polymorphism in patients affect response to the TRIKES?
I don't know that. It's a good point. I just don't know. I just don't know. We didn't essay that at all. So I just don't know.
Difficult experiment to do because you will need a large cohort and you need to. There is also a question about the immunogenicity of these TRIKES. Do you think they can be immunogenic and therefore at some point detrimental or less efficacious?
Yes. So as you know, the immunogenicity of therapeutic antibody is not predictable. I should say that probably, as everybody knows, there is no difference in immunogenicity between humanized antibody and fully human antibody to begin with. So at each time you have a new therapeutic antibody, you have a new generation of ADA, and you just cannot predict that. So we don't see any warning for that. But I should have said probably that the NKCEs are running for IND, but they are not in humans yet. But we don't expect any kind of warning regarding ADA, not more than any other therapeutic antibodies.
Okay, so there is another question from Virginia Maria Vlashava asking, would you expect the TRIKE to cause a cytokine storm or autoimmune disease, especially the one containing IL-15?
Okay, so we are not working with the one having IL-15. These TRIKEs have been generated by Jeff Miller. His group didn't report any kind of any signs of toxicity so far, but I'm not aware of all his data. We don't see for ourselves in this version of NK cell engager that we have presented today why we should induce autoimmunity. I would like to emphasize the fact that injection of allogenic NK cells, hyperactivity allogenic NK cells in patients was never associated with any kind of toxicity so far. So I don't think that toxicity is a warning for NK cell-based therapies.
And there is also a question about what is the lifespan of NK cells? What is the NK cell count in healthy and cancer lesions? And do you worry about the fact that there are not enough NK cells in cancer lesions?
Yeah, sure. So the half-life of NK cells in vivo is always a matter of debate. The rough estimation will be around 2 weeks for the half-life of NK cells. As I said, by harnessing NK cell function, we are not hoping to debulk the tumor. What we are hoping to do is to contribute to kill some tumor cells, but maybe much more than that, to provide the spark that will initiate a multicellular immune response. That's why we are so interested in initiating an innate immune response and not being only T-cell centered. So I don't see why it should be a limitation, even though NK cell might be short-lived.
Yeah, absolutely. So I'm going to, you had so many questions. I'm quite, this is extraordinary. So the audience is very engaged, likely because you were very engaging. So a question about the second part of the talk, so the COVID, which I find very, very interesting. And just one comment, Eric. So this IL-16, which IL-6 contribution to disease, which I believed on, initially, I totally agree with you. I think it's probably overestimated or not as potent because most of the IL-6 blockade trials didn't pan out. So either because it's not the only driver, or maybe because it's not the main driver. And your work on C5a is really, I think, spot on. So there are many questions about this, and one question was, what is, in your opinion, this cause of lymphopenia Maybe this is not the best. Well, yeah. Don't know
Yes,
whether
well,
you study that, but
I
what's
really
the cause
don't
of
know.
lymphopenia?
I should ask you, Miriam. Do
Yeah.
do you know?
Yeah.
Frankly,
I don't know.
I don't
Yeah.
know.
Yeah. No, we don't know either. It's a we don't have much time. This is a very important question. I think we just
Yeah, sure.
don't know why, but it's very clear. Everyone saw it. There is a question on, do you think there is any evidence that NK cells may contribute to the killing of T cells and B cells in this severe COVID infection?
In the context of severe COVID-19, I don't know. But what I know is yes, NK cells can kill activated NK cells, activated T cells, sorry. That's for sure.
Yeah.
We know that since 30 years. The original data on the identification of MHC class 1, not restriction, but inhibition of NK cell function was based on the fact that people were using the PHA blast as NK cell targets. So yes, NK cells can kill activated T cells, and we're actually collaborating with Annette Oxenius on that, because NKp46 is really involved in the killing of activated T cells by NK cells, by the way.
Yeah. So the other question is, do you think C5a is responsible for ARDS beyond in patients without a cytokine storm? And does it have a role, in other type of ARDS beyond COVID?
Absolutely. Actually, we started from there. When you look again at models of influenza infection that lead to ARDS, we see people have seen a clear involvement of C5a. Yes, so that's for sure. Now, the question that we are asking is whether C5a is involved in COVID-19, and only the clinical results will tell us whether it's only a marker or it's an actor. We just don't know.
Yeah, or the problem with these trials, Eric, is that we are blocking, one event very late on, so it can still be an actor, but not sufficient. But the blockade is not sufficient to really improve disease severity, right? So
you're absolutely
I hope
true.
You guys are doing some immune. Yeah,
you're absolutely
hope
true.
You are doing some immune monitoring. Yeah,
and that's why it's interesting to think of the timing of administration of avdoralimab. Which is a very well tolerated molecule, by the way, which is totally in contrast with eculizumab, because people have been trying to block C5, and we think that this is putting people at risk, patients at risk, because we need C5b for the formation of the MAC and for control of bacterial infection. So blocking C5 is not an option for us. We really do prefer to block C5a or C5AR1 in our case. To leave the MAC totally intact. And as the molecule is well tolerated, maybe it would be important to treat patients very early on with avdoralimab if there is a sign in the fourth clinical trial.
This is a great answer. In fact, there was a question about eculizumab. As you know, in our centers, we hesitated, and eculizumab was advanced. I totally agree that blocking blocking C5 is dangerous. In fact, most of our patients that were intubated at some point had bacterial infection. In fact, all of them. This co-infection is so well described. So your approach is really very interesting. There is the last question, maybe, because I have hints that we need to stop soon.
Okay.
Do you think that innate lymphocyte or NK cells are playing a role contributing to COVID-19 disease severity?
I don't know. I just don't know. Frankly, I don't know. So there is lymphopenia. We've been trying to look at ILCs, but it's very difficult because ILCs are not much present at all in the blood. And it was very difficult to have access to tissues for obvious reasons. So we just don't know. It's really a question mark. But before we stop, I would like to thank everybody again. And I would like also to say that people who have been asking questions that I could not answer, please send me an email. I will be much happy to answer these questions.
So maybe I'll ask one last question about ontogeny, because this is very new and very interesting. So I just for clarification. So you think there is this fetal liver recruitment of progenitors in the tissue that will persist for a prolonged period of time? This is what you think?
During adulthood, yes.
During adulthood, but I still didn't understand the gamma link. So at some point there is a differentiation with the to ILC1, which is independent of NK, completely different, right? So they differentiate into ILC1. If you can clarify, for example, you went a little bit fast. So recruitment, they persist, they differentiate, they still need external cue, which you suggest is coming from the differentiated cells. So I lost that. Like how?
Yes,
How they?
I recognize I was going very fast. There are, we hypothesize there are some environment cues in the liver which are inducing interferon gamma production by ILC1s. And this interferon gamma produced by ILC1s in the liver is amplifying the development of liver ILC1s. So I think that it's important because it comes with the idea that there is a regional immune response, that the immune response of the liver will be different from the immune response elsewhere. And so we just don't know what are the liver cues that induce gamma interferon by ILC1. That we don't know. But again, I think it's close to what you have done in the myeloid system, showing that the macrophages would develop there, and from embryonic progenitors, we do think it's exactly the same for ILC1s in the liver.
And so do you see, this is very important, because I think that the shaping of these cells that persist for prolonged period of time in tissue, shaping by tissue cues, is going to provide a cell that's quite unique because of all the signal that they receive. And it's very difficult. So when there are new ones that come, maybe in response to some injury signal, they have different role because, they were educated differently. So do you see that? Do you see ILC1 in different tissue having different molecular program?
No, we don't see that. We don't see that yet. We are studying that. What I can tell you is that the loop that we are describing here is only working in the liver. For some reason, when you look at ILC1s in the small intestine, for example, you really don't see the loop that we have been documenting in the liver. So there is something special in the liver. So which is not only special according to the other ILC1 population, but also very special as compared to NK cells, because NK cells in the liver are not coming from the same route. They are coming from the bone marrow, which is not the case for liver ILC1.
That's interesting. This is the case for mast cells also. Mast cells in the skin, for example, are maintained locally. Mast cells in the mucosa, in the intestine, are constantly repopulated by circulating progenitors.
Interesting.
There is so much to discover about this innate system. We spent a lot of time on the adaptive system, which is great. We have to focus on the innate compartment, right, more.
Absolutely.
Which is, So thank you very, very much, Eric.
Thank you,
This was a very interesting talk, fantastic, different aspect of the immune response, highlighting how much immunology, can be used, clinically. Thank you. So we are now going to stop here. If you have questions, please feel free to reach out to Eric. As I said, he will take the time to respond to you. Don't bombard him too much, but I'm sure you'll take the time. These were very good questions. I mean, the audience was really, really on top of everything. Anna, do you want to put your last slide, the slide that you mentioned here? I just want to say that we hope that you will also be attending the next seminar, which will be given by Joanne Flynn, a fantastic investigator from U Pittsburgh, who is part of the Vaccine Research Center at University of Pittsburgh. Thank you very much for your engagement today, and I wish you all very well. Please be safe.
Bye-bye.
Bye-bye, everyone.
Thank you.